Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. So today we will discuss about the fat stage and physiology of the fat stage. Not biochemistry but physiology of the fat stage. Next video we will discuss about with the biochemistry. So here is the fat state is related to the glycogenesis. It means the glycogen will be formed into the liver and muscle, and lipogenesis mean the fatty tissue, which that is adipose tissue, will be formed and fed the lipids and energy stores, and glucagon decrease, which that is the hormone, and insulin increase from the beta cell of pancreas, and anabolism will be possible and catabolism will less. Anabolism means the making of new molecules, and catabolism means the breakdown of larger molecule into the smaller molecule. So the glycogenolysis will be not formed, but glycogenesis will be occur. So here I draw I have drawn the blood vessel, which that's contained here is the first of all we will discuss about the stomach. So first of all, salivary gland, which that is created amylase enzyme, salivary amylase enzyme, which that break down the partially starch, and that's go into the stomach in the form of bolus, and the stomach will digest the protein, and that protein will be partially digested into the polypeptides of protein. So that will be polypeptide of amino acid that will be converted into the chyme, and this chyme is the very important about that physiology. So here is the duodenum, which that's contain enterocytes. So these enterocytes contain character is brush border enzyme, so that is also known as the brush border cells. It is also contain a microvilli, so these microvilli increase the surface area. So here is the pancreatic amylase, which that is further digest the disaccharide and oligosaccharide. So these will be sucrase enzyme also present in the brush border enzyme. That's sucrose breakdown into the simple molecules. So that's all thing is related to the digestion. So that's digestion will be occur, and that's sodium move inside to create a one molecule allowed to move inside of the glucose. That is the GLUT2 transporter, the GLUT2 channel, which that is created is here, and here is a GLUT4 channel, which that is allowed to move inside into the blood. So we have covered the basically um, mm, basically carbohydrate digestion, and that's going to the blood. So next is the here is the protein, which that is made up of N-terminal and carboxylic terminal and amino terminal, which that is basically nitrogen terminal and carbon terminal, which that is a polypeptide digested by the produced enzyme from the pancreas, trypsin and chymotrypsin. These trypsin and chymotrypsin will be further break down this polypeptide into the dipeptide, tripeptide and amino acid as well. So that is the will go inside by the same mechanism, but here is the 2 sodium ion move inside to create a one allow to one molecule of the amino acid or disaccharide, which that is the basically we are basically understand about the moving the amino acid. So next video we will discuss about the digestive system, and that digestive system will contain this information. So here is the sodium ion and potassium ion by the ATP used to produce one amino acid move into the blood. So that's moving of the blood to inside. So what happened about it? So these amino acid will be increased into the blood vessels, into the circulatory system. So what will be happen, that is the protein basically um, used into the liver. So here is the fat, which that is basically, here is the tripeptidase and dipeptidase enzyme on the present on the brush border cell, which that is also um, breakdown of the of the peptide bond, and that's converted into the amino acid. On the other hand, more and more, here is the fats, which that is basically convert into the micelles. Is micelle made up of phospholipids, triglycerides, and monoglycerides as well. So that is the lumen, which does contain a fat, and this fat will be go into the enterocyte and first move into the lymphatic system. But here I will not draw a lymphatic system. Lacteal will not draw here. So that is why, just for understanding about the mechanism. So these micelle will go into inside, and Golgi apparatus will pick up the apoprotein from the enterocyte and some fats to modify it to make a little bit protein is known as chylomicron. This chylomicron is known as the very low density lipoprotein, and these contain a lipid and protein. But protein is very minimum. It means that 10% is protein. And this chylomicron is the very special character which that is used for the anabolic effect during the fed stage. So this fed stage is, for example, here in go into the blood vessel. So that's here is the blood glucose will be rise during fed stage, and amino acid will be rise. It means the amino group will be rise. So these amino groups or amino acid will be move into the muscle for using making a new cell repair, cell division, and muscle fiber making after the workout. So here is the chylomicron fats rise. So these chylomicrons, which that's lipase enzyme used into the blood vessel to produce glycerol and fatty acid, which that move into the adipose tissue is here, and this adipose tissue convert into the glycerol, triacylglycerol, and this is adipose tissue. On the other hand, the rest of the tissue which that are present in our body is the hair, which that is cells show of this tissue, etc., etc. For example, that tissue can be muscular, digestive system, or brain, etc. So these also use the fatty acid for further produce energy, either go into the adipose tissue or go into for using as energy. So here is the liver, which that is I have drawn. So these will be basically is made up of gallbladder, and that is the two lobe is the left and right. It means the right lobe is basically larger and left lobe is the smaller. So this is the remnant very low density lipoprotein. For example, chylomicron will go into the liver by the LDL receptor. So the endosome will be produced, and that will be different enzymes used, lipase and protease. That's all enzymes used to degrade it, and this produce triacylglycerol, fatty acid, monoglycerol, as well as glycerol. So monosylglycerol, etc. So on the other hand, there is protein which that's amino acid come from, and here is a modified into the liver to produce apoprotein A, B, C, and D, E. So these will be cholesterol also formed from the glucose, and that's fatty acid and triacylglycerol will be formed by the malonyl CoA, and that's phospholipids also produced from the which that is dietary phospholipids, which that's go into the Golgi bodies, and that's modified into form a high density lipoprotein, which that's fill and contain protein. These go into the high density lipoprotein into the blood. So it means the high density lipoprotein will be go into the blood. If too much fats are present into the body, so the excess fat will release from the bile duct, from the gallbladder, which that is bile will excrete that excess of cholesterol, bad cholesterol, low density lipoprotein. So here on the other hand, the beta cell which that is stimulated due to the amino acid and glucose increase, fat will not trigger the insulin production and glucagon will release and increase. So here is the pancreas which that is contain acine or island of Langerhans, which that is important character is that alpha cell, beta cell and delta cell will produce. Alpha cell produce glucagon, beta cell will produce insulin and delta cell will produce somatostatin. So the insulin released from the beta cell is basically the group of cell play endocrine role for metabolism. So these enzyme this hormone is used as an endocrinal system. Endocrine system, it means the go into the blood, not go into the exocrine which that is for example pancreatic amylase. So here is the glucose which that move into the tissue, and that's basically go by the releasing of insulin binding to increase that is basically glycogenesis into the muscle. So the glycogen will be formed, or either go into the glycogenesis or go into the glycolysis to produce pyruvate, and these pyruvate go into the acetyl CoA into the mitochondrial inner matrix, and dry carboxylic acid will be formed, and this dry carboxylic acid cycle will be formed to produce carbon dioxide as well as energy, ATP. More and more, here is the mitochondria, and here is the triglycerides will be produced also from mitochondria, which that is a malonyl CoA will be formed. So just remember about these things, and that's going into the blood vessel is known as a very low density lipoprotein, and these very low density lipoprotein transport into the adipose tissue and fatty acid which that also sugar which that is also convert into the glucose in from into the adipose tissue by the by the lipogenesis from the glucose. So that's all thing is anabolically active and working. Still here is the glucose which that's move inside to the liver, and that is also insulin like growth factor which that is bound by the growth hormone, and that's produce glycogenesis or lipogenesis, either go into the Krebs cycle. So the glycogen will be formed into the liver. On the other hand, here is the sugar which that is also go into. So the brain cell which that receive the glucose by the glut channel obviously, and there is a glucose transporter which receive that is glucose inside by the active transport and produce pyruvate and acetyl CoA will be formed, and carbon dioxide and ATP will be produced. On some same mechanism is here, which that is the glucose will move inside of the muscle, and there's the glycolysis will be formed to produce pyruvate and acetyl CoA, or go into the glycogenesis. So that's different mechanism. So glycogenesis will be formed and glycogen will be formed. On the other hand, the glycolysis produce acetyl CoA, and that's ATP and carbon dioxide. On the other hand, here is the red blood cell which that contain a pyruvate which that is produce a pyruvate and lactate which that's convert pyruvate to lactate, not acetyl CoA, because mitochondria is not present. So the glycolysis will be just occur and red blood cell will be form is here. So this mean is here is the acetyl CoA will be formed, and that is the basically RBC which that is release the pyruvate into the muscle cell or other cells. So this all thing is related to the fat state. So here is the RBC will produce pyruvate, and basically convert into the lactate, and that's lactate and pyruvate also go into the muscle cell, either go into the muscle cell or use as a basically energy is just on the glycolysis in the RBC. So thanks for watching.